the right things on the right side and the wrong things on the wrong side.
Cosine equals 87 divided by 105, the square root of 105.
I mean, I can't always be their little girl.
The way of the world. One life begins with two. The seed of man joins the egg of woman, and the essence of both mingle and grow. In the warmth of the uterus, the new entity is nourished, allowing the miraculous process of development to flourish. Cells multiply and transform into a child with a primitive heart, pulsing the rhythm of life. A system of support and protection emerges. The growing embryo forms eyes that will see, legs that will run, arms that will hold others in need and in love. In only weeks, the entity takes on a recognizable human form. Features and organs become more and more refined, as fetal size surges from under an ounce to pounds. In the last months, the sex of the developing child becomes evident. Male or female, it's a feature that will shape much about his or her future life. The moving, aware being makes its presence increasingly known to the mother. Nourished by her in the warmth and darkness of the womb, until the moment arrives when a new child crosses the threshold into the light.
Thank goodness. It's a boy.
It's a boy.
You bet it's a boy. What's his name?
Sean Patrick.
Okay, and then we're going to give you right over to Mom.
"30 years ago, Julie came into the world appearing quite normal. Soon, however, a genetic disorder left her with permanent brain damage. Chase, born with the same disorder, is developing normally, thanks to recent discoveries that have unlocked many of the mysteries surrounding the interplay of environment and genetics. A key discovery has been in our greater understanding of the molecule of life itself, DNA, deoxyribonucleic acid. In 1953, James Watson and Francis Crick discovered the spiral staircase structure of the DNA molecule, the basic unit of heredity.
What strikes me about DNA is it's the end product of 4.5 billion years of evolution. And evolution has carried out this magnificent feat of making this one-dimensional code that contains all the information that allow us to start out as a single cell and develop into a full adult organism.
The human cell contains genetic information from both parents' DNA, arranged in pairs of chromosomes.
The 23 pairs of chromosomes that are present in each and every human cell are essentially the blueprint for life. So they have all the information that's necessary for starting you out as a single cell and carrying you through this marvelous process of development where you end up being an organism with 10 to the 14th cells. That's a 1 followed by 14 zeros, an astronomical number.
We're constantly making new cells. Cells are dying, and we're making other new cells. In a developing embryo, the embryo is gaining size as cells continue to multiply and divide. At a certain point of cell division or mitosis, that's when differentiation occurs. That's when some of those cells are going to develop into heart, and some into stomach, and some into brain and central nervous system.
Genes, segments of DNA, dictate to the cells how to make a human being.
We got weather.
Even specific traits like Robert's red hair, Timmy's blue eyes, and Julia's brown skin are passed from parent to offspring. However, traits may skip generations. We can see how this works by following the genes of brown-eyed and blue-eyed parents. The two large bees represent the brown eyes gene. The two small bees, the blue eyes gene. When a child is conceived, the parent's genes separate and recombine. So each parent contributes 1/2 of each child's genes. For this family, brown eyes prevail in all children. Even when the blue eyes gene is present, the brown eyes gene dominates. However, this doesn't mean that blue eyes are headed for extinction. Watch what happens when brown-eyed children carrying blue eyes genes later become parents. The recessive trait now has the chance to reappear as the genes separate into 4 possible combinations in their children. Chances are 3 of the children will have a dominant gene leading to brown eyes, and only 1 in 4 both recessive blue eyes genes, allowing blue eyes to appear. The odds for a blue-eyed child grow when one parent has brown eyes with a recessive blue eyes gene, and the other parent has blue eyes, that is, 2 recessive genes. When their genes separate and recombine, the chances become 50-50 that the recessive trait will appear. Eye color is just one example of the thousands upon thousands of traits we possess that help to make each of us physically unique. Unfortunately, a genetic inheritance can also include a recessive disorder, like phenylketonuria, or PKU, which unchecked allows large amounts of phenylalanine to build up in the body.
Phenylalanine in high concentrations is toxic to the brain, so that children who have PKU who are untreated will be profoundly mentally retarded. Usually they'll have IQs less than 40, so very profoundly retarded.
Fortunately, PKU can be controlled by determining through trial and error the amount of protein each child can tolerate in his or her diet.
Meat, what? You can't have cheese and no eggs, right? We are manipulating the environment to fit with the unusual genetic manifestations in the child. If we make sure that there is not a toxic amount of phenylalanine in the diet, we can assure a normal development of the brain in children with PKU.
This intervention technique was not available when Julie was a child. More recently, however, the trial and error method of determining protein tolerance has saved many. Hoping to further unlock the mystery of this genetic disorder, Dr. Smith initiated a DNA study of her patients.
The purpose of the threshold study is to find a more accurate way of determining how much protein a baby or child can tolerate without developing high levels of phenylalanine.
To conduct such genetic research, Dr. Smith and her associates take samples of each patient's blood, extract the DNA, and begin the lab-intensive process of searching for differences between those with high and low protein tolerance. Locating the genes associated with a disorder would be an impossible task for researchers without the aid of genetic maps.
Thousands of different genetic diseases are known to be genetic because they're inherited, but we have no idea what the abnormal enzyme or protein is in those diseases. If we can map the inheritance of that gene, in other words, map the genetic trait to a specific human chromosome, this opens the way for us being able to isolate the DNA for the disease gene even before we know what the protein is.
For many, a better understanding of the interplay of genetics and environment offers hope for the future.
It's absolutely incredible to think of how much genetic variability two of us can produce in creating offspring and in the kinds of things that might have happened to that genetic material that would have implications for later in development. Anything that might happen that would produce genetic damage early on that we would then see the consequences of later in development.
Tragically, certain substances collectively called teratogens sometimes distort the course of development. Cigarette smoking, drug use, or drinking by the mother can damage a developing embryo or fetus in the womb. Even small amounts of such substances can be devastating.
Apart from causing the abnormal development of the brain and the heart, we notice that babies exposed to alcohol during pregnancy have a very low birth weight, and low birth weight in itself frequently causes complications in the early postnatal period. The fetal alcohol syndrome is widely recognized because of the small head size and abnormal facial features. But scientists are becoming more aware of another aspect of this problem, which is referred to as fetal alcohol effects. That is, there are children who do not show the facial features of fetal alcohol syndrome and who do not have the small head size, but nevertheless seem to have abnormal central nervous system development, which causes them to have behavioral problems and learning defects.
Pregnant women from all walks of life use drugs and make other lifestyle choices that may be harmful to their developing child.
We have women who come to us from the inner city, poor, Hispanic, black, white population. We have women who come from the suburbs and from the posher neighborhoods who have used drugs in a recreational way, as they call it, at parties and social gatherings. These women all have one thing in common. They have used drugs during pregnancy.
One of the things that we know is that if women use cocaine during the first 3 months of their pregnancy, that they are more at risk for spontaneous abortions. Okay? You understand that? Yeah. Now, as far as the pregnancy later on, we see that if women continue to use cocaine and use during the last 3 months, that they are at risk for early delivery, going into delivery early, premature labor, and so that's very dangerous.
One of the things that we see about babies who have been exposed to cocaine is they tend to be very tremulous and shaky, and a lot of times you'll see the baby suddenly start shaking like this, very fine kinds of tremors. Also, they're very easily disturbed. Just picking them up very gently a lot of times will set them off. I'm going to take the pacifier out a second. So you can see that little fine tremor. That's the kind of thing we're looking at. Now, the baby was only about 5 pounds, 13 ounces, which is kind of small for babies. A lot of the babies exposed to cocaine are quite small. And we think that that's related to the use of the drug during pregnancy.
Some disorders occur despite the best intentions and efforts of the mother. However, the damage caused by teratogens is in most cases predictable and avoidable. Every prospective parent hopes for and expects a healthy child.
You want me to go though?
However, many are concerned with the possibility of birth disorders because of race, ethnic background, family history, or age of the mother.
There are some people that have genetic diseases in their families, and they're concerned about what the risks are for them having children with the same type problem.
Among birth disorders feared most are Tay-Sachs, PKU, sickle cell anemia, and Down syndrome.
The vast majority of patients that we see here has to do with the women that are over 35. And when a woman is over 35, her chances are higher that she might have a baby with a chromosome abnormality such as Down syndrome.
Well, most people are familiar with Down syndrome, and it's a situation in which there is an extra number 21 chromosome. So instead of having 2 of each pair, there's a third of the number 21 group, so that there are 3 number 21 chromosomes, and that is the basis of Down syndrome.
Kathy, I see my nephew that has PKU, and it concerns me. What is our risk of having a child with PKU?
Because your brother is a carrier, and because we know that brothers and sisters share half of their genetic information, your chance for being a carrier is 50%. None of us are immune from genetic diseases. All of us have about 5 to 8 genes that are capable of causing disease. The reason why we don't see the disease as frequently as one would expect is that the likelihood that 2 people who come together and have a child together, the likelihood that they would both carry the same gene for the same disease is still fairly rare.
One of the things that I've always found amazing about development has been that there are so many places where things can go wrong, and it's surprising that things don't go wrong more often than they do, and that the majority of babies, in fact, approximately 97% of all newborn babies are normal, at least as can be determined at birth.
Is there anybody in either family of Jewish ancestry that you know of?
My grandfather is Jewish ancestry on my mother's side.
Okay. Actually, it seems like every ethnic background has some sort of a genetic problem associated with it. The more common ones that we're aware of are people of Eastern European Jewish heritage being at higher risk for a disorder called Tay-Sachs disease. People who are black are at a higher risk for sickle cell disease.
Disorders such as sickle cell anemia and Tay-Sachs are not limited to commonly known high-risk groups. Fortunately, prenatal testing can help identify some potential problems in advance.
When you do prenatal testing, there's 2 kinds of tests that we can do. The first one, which is the one that's been around the longest, is amniocentesis.
When can we do this test?
This test is done later. This test is done between the 16th and 18th week of pregnancy. There is another test that we do called CVS, chorionic villi sampling, that can be done earlier. It's done between the 10th and 12th week of pregnancy. There are probably about 400 different genetic diseases that we have the capability of diagnosing by using tests such as chorionic villi sampling or amniocentesis. A lot of those cannot be diagnosed routinely in families because you need to have an affected member of the family before you know that there's a risk. We don't routinely test for all of those things in a couple that would come in without a family history.
Kathy, what has a higher risk, amniocentesis or CVS?
At age 34, you're what we call borderline in terms of your risks for having a baby with a chromosome abnormality. Do you ever wonder why 35 is always the number that you hear in terms of how old a woman is when she should have prenatal testing? Mm-hmm. The reason why 35 is the age that we use is because at age 35, the risk for a chromosome abnormality is 1 in 200. That's the risk also of having a problem, a complication, such as miscarriage, that would result from actually doing the procedure.
In a small percentage of cases, testing can threaten the survival of a growing fetus. That possibility makes the choice difficult.
You'll notice is 1 in 300.
If parents do decide to test, a lab takes a specimen from the mother's womb. Chromosomes are then extracted from processed cell samples and stained so they will show up clearly when photographed. The results are then pasted up in an arrangement called a karyotype, ready for analysis.
When I look at a karyotype, the first thing that I look at is the chromosome number, to be sure that the proper number of chromosomes are there. And there should be 46 chromosomes arranged in 23 pairs. The next thing that I do is to look at each individual chromosome and to make sure that it matches its sister chromosome in each pair, to be sure that there are no structural rearrangements, and then more specifically to look at the chromosome to make sure that all of the recognizable bands are visible and present and in their proper location. When we treat the chromosome with a stain, certain portions of the chromosome will take up the stain differently than other chromosomes, and typically there are areas that stain darkly and areas that don't stain very well, and in that situation you will result in a differential banding pattern, so that there are a series of light and dark bands.
Discovery of a genetic abnormality answers one question, but raises many more.
Well, the problem with disorders such as Down syndrome are that the effects can be variable. There's a spectrum of abnormality. It is fair to say that virtually all individuals with Down syndrome will be mentally retarded or will be slow. However, there are some very high-functioning individuals who may test with IQs near the normal range or even rare occasions in the normal range.
Well, it's great to see you both again. And as I mentioned on the phone when we talked, the chromosomes were normal on the preliminary part of the test, and the finals, they're also normal. Everything looks good. The baby is a boy.
That's exciting. Well, it's exciting for him. Well, it's exciting for both of us, but he's the one that really wanted a boy.
Let me show you what your little boy's chromosomes look like. You'll notice again that there's 2 of everything. You'll notice that there's an X and a Y. Everything's present. There's nothing extra, nothing missing, nothing changed around. Everything's looking real good.
Hi, baby.
Imagine a world where each child's genetic destiny is totally controllable, ensuring good health and life without disease or disorders. Some believe this fantasy, if it could ever happen, would take centuries to accomplish. Yet, faster than anyone ever imagined, fantasy is becoming reality in labs around the world. In the 1980s, Dr. Leroy Hood helped launch a decades-long effort to identify the estimated 50 to 100,000 human genes. The first step in making such a future a reality.
The Human Genome Project is about deciphering the human genome, that is our 23 pairs of chromosomes. What's exciting about the goal, really, in my view, is 2 things. To do this, we're going to have to develop very powerful technologies for mapping and sequencing the human genome, and new computer technologies that will allow us to understand this information. And these new technologies will revolutionize biology and medicine.
At the California Institute of Technology, Dr. Hood and his associates are developing tools to more rapidly read the DNA alphabet, defined as base substances abbreviated as A, G, C, and T.
There's 3 billion bases inside every human's chromosomes, and we want to determine that sequence. The sequence of the 4 bases, A, G, C, and T, on the chromosomes inside your body are essentially a computer program that determines whether you're a human with a disease, blonde, blue-eyed, whether you're a mouse or a horse. And our goal is to determine the sequence of those bases as rapidly as we can.
To help accomplish this enormous task, technicians tag the DNA bases with fluorescent molecules of different colors so they can be rapidly scanned by a laser, which then feeds the information into a computer. Each band of color represents different letters of the DNA alphabet. The computer then translates these colors into a printout of a DNA sequence.
The exciting potential applications of the Human Genome Project over the next 25 years are many. One that excites me in particular is the idea that in that time we'll have identified perhaps 100 genes that predispose us to various types of disease. So at birth we can take DNA from a newborn, we can carry out the DNA fingerprints, we can put the information in a computer, and On the one hand, the susceptibilities to disease will be read out, and on the other hand, the computer will tell us what therapeutic intervention measures will be necessary to circumvent any limitations that those genes may have. So we'll be able to use this information to ensure most humans lead a healthy, mentally alert life right up to the end of their natural lifespan.
For tomorrow's children, the impact of this quest to reveal our biological secrets will be enormous, but not without controversy.
In the future, we'll be able to engineer humans. We'll be able to trade bad genes for good genes, or we'll be able to possibly change some of the very fundamental traits of human organisms. And all of these issues must be considered carefully by society so guidelines can be set as to how we use the information. To argue that the information is too dangerous and that we shouldn't generate it, I think is irresponsible, because it will change the lives of literally millions of people when we can cure them from the diseases that genetics have generated. On the other hand, we as a society have to learn how to deal with these technological problems and answer in a satisfactory way the social, ethical, and legal challenges they pose.
From the moment of conception, our journey to birth is directed by a delicate interplay of our genes and the environment in which we develop. The hazards of that journey can include a host of disorders and diseases, some avoidable, some not. Yet, our emerging ability to control the course of development brings new hope, together with new choices and new responsibilities. And regardless of scientific breakthroughs, ultimately our destiny lies not in our genes, but in ourselves.